Hello, my name is Sungwoo Bae from Institute of Radiation Medicine in Seoul National University. In this 20 minutes talk, I would like to suggest a method which integrates spatial transcriptomic data with unmatched single-cell data, such as sorted single-cell RNA-seq data to obtain spatial distribution of a few cell populations. Before I go on with the details, I'll briefly explain the importance of investigating spatial context of cells within the tissue. The cells that make up the tissues do not act alone, and they come into contact with adjacent cells or interact through ligand-receptor connections. The interaction can change according to the spatial distribution of cells. Thus, it is a crucial task to understand the spatial configurations of cells within the tissue. The spatial distribution and characteristics of cells are essential not only in homeostatic conditions, but also in the differentiation process of cells and in pathogenesis. For example, the spatial compositions of tumor cells and immune cells in the tumor microenvironment is a key to comprehending the spatial dynamics and evolution of cancer. One of the effective platforms for understanding the spatial characteristics of cells is barcoding-based spatial transcriptomic data. Recent advances in technology have enabled investigations of tens of thousands of RNA expression corresponding to each location and tissue using the spatial barcodes. The gene expression profiles obtained in each spot, the basic unit of spatial transcriptomics, is mapped to the tissue, and spatial map of genome-wide RNA expression can be acquired. However, the spatial transcriptomics has a problem in that spatial information cannot be obtained in the units of cells because the boundary of the cells and the spot does not match. Several cells or part of the cells can be contained in one spot, and the density of cells vary across the spots. The problem can be overcome with the advent of model that integrates single-cell and spatial transcriptomes. Since single-cell data explains the gene expression in the unit of cells and spatial data in the unit of spots, the arbitrarily divided tissue domain, one can think of integrating both datasets to decode spatial composition of the cell types. The integrative methods are based on an assumption that cell types are similar between single-cell and spatial datasets. First of all, cell type should be defined from the unsupervised clustering of the single-cell data based on gene expression. After the annotation of the cell types, the cell type markers and expression profiles were utilized to deconvolute the fraction or abundance of the cell types in spatial data. However, there is a limitation in the existing deconvolution methods, particularly when cell types composing the single cell and spatial data are largely different. In case of investigating infiltrating cell populations such as immune cells, single-cell RNA sequencing is commonly combined with cell sorting strategies to characterize a few cell populations. Since the single-cell data explains only the part of cell types in the tissue, it is difficult to accurately capture expression signatures of the cell types from sorted single-cell dataset and transfer the information to the spatial data. Therefore, there is a need for a method to flexibly integrate the sorted single-cell with spatial transcriptomic data to study a spatial context of minority cell populations. Here we suggest a method named as spSeudoMap to spatially map the cell types of single-cell data acquired from cell sorting experiments. The subpopulation single-cell data are composed of sorted cells from the tissue, and the cell types cover only part of those in the spatial transcriptome. To create the reference dataset that mimics the spatial data, the virtual cell mixtures named modified pseudospots were defined in which all cell types from the tissue were included. First, cell types exclusively present in the spatial data were aggregated and named pseudotype. The virtual markers for the pseudotype were selected from the top genes highly expressed in spatial pseudobulk compared to the single-cell pseudobulk data. Then the pseudotype fraction in the spatial spots were estimated from the module scores of the top 20 virtual pseudotype markers. The fraction and gene expression of pseudotype were assigned based on the presumed pseudotype fraction and expression of the randomly selected spots from the spatial data. Last, the non-pseudotype proportion of the modified pseudospot was filled with the randomly selected cells from the subpopulation single-cell data. Finally, the modified pseudospot was considered as a reference dataset for domain adaptation method CellDart. When breaking into the details, the core parts of the spSeudoMap are predicting pseudotype fraction in spatial transcriptomic data and assigning synthetic gene expression profiles of pseudotype in virtual cell mixture. The key parameters of the model can be summarized into three aspects. First, mean and standard deviation of the pseudotype fraction in the tissue. Second, number of markers per cell types in single-cell data. Third, the ratio of number of single-cell markers to pseudotype markers, MK ratio. In the first part, virtual cell type markers were extracted from pseudobulk count metrics by log-fold change, and the module scores were calculated. The module scores represent the average expression level of the gene set compared to the randomly selected gene pools. The module scores are expected to reflect the proportion of pseudotype in each spatial transcriptomic spot. The module scores were linearly transformed to have a certain mean and standard deviation values. The mean of pseudotype fraction was determined by literature evidence or cell sorting experiment, which informs the average proportion of non-sorted cell type fraction in the tissue. The standard deviation of pseudotype fraction over 0.05 brought us the optimal performance. As a result, pseudotype fraction was estimated in all spots composing the spatial transcriptomic data. For the next step, the spots were randomly selected from spatial data and presumed pseudotype fraction and expression profiles of single-cell markers and pseudotype markers were extracted. Since the expression of pseudotype markers and pseudotype is expected to be significantly higher than that of single-cell markers, so for the simplicity, the single-cell marker expression in the pseudotype was set to 0, and the pseudotype marker expression was assigned by multiplying the expression in the selected spots by the presumed pseudotype fraction. Then the non-pseudotype proportion of the modified pseudospot was filled with the randomly sampled cells from the subpopulation single-cell data with random weights. Lastly, composite gene expression profiles and the cell type proportions for the virtual cell type mixture were calculated, and dataset were named modified pseudospot. The last process adopts CellDart, which performs domain adaptation between the modified pseudospots and spatial transcriptomic spot. In the point of the dataset, the neural network first learns to calculate proportion of cell types from the gene expression of the modified pseudospot. It is a supervised process with the known cell type proportion. And the neural network is modified by adversarial domain adaptation process, such that the model cannot distinguish between the pseudospot and the real spots in the latent space. Finally, the fully trained neural network can precisely predict the cell type compositions both in original pseudospot dataset and in the real spatial transcriptomic dataset, in which the cell type proportion is not known. The datasets were utilized as in the table to validate the suggested tool and apply the method to the real sorted single-cell dataset. First of all, we wanted to evaluate the performance of spSeudoMap and existing tools when integrating subpopulation single-cell dataset directly with the spatial transcriptomic data. The simulated subpopulation dataset were generated by extracting cortical layer-specific excitatory neurons from single-nucleus dataset of human brain tissues. We compared the performance of spSeudoMap with cell2location and CellDart both of which showed comparable performance in our previous research. The performance was measured qualitatively and quantitatively by calculating layer discriminative accuracy of predicted neuron fraction. Next, we validated spSeudoMap in mouse brain coronal section dataset. The region-specific excitatory neurons were extracted from the single nucleus dataset of mouse brain to generate simulation dataset, and spSeudoMap was applied to predict the spatial distribution of neuron types. This time, whole cell types in the single nucleus dataset were mapped to the tissue using cell dart and cell to location. The results from the two methods were considered as references to evaluate the performance of spSeudoMap. Finally, we applied spSeudoMap to estimate the immune cell compositions in breast cancer by utilizing CD45 positive sorted single cell dataset. The results were compared with the reference, applying the cell dart, which mapped whole cell types in the unsorted single cell dataset of the same patient. First, spSeudoMap was evaluated in human brain tissue. Among the various cell types present in the single nucleus dataset, only the 10 layer-specific excitatory neuron types were selected, and they were utilized as a simulation dataset for the sorted single-cell data. When these 10 excitatory neuron types from single-cell data were spatially mapped to the human brain using spSeudoMap, the spatial distribution of the neuron types were highly restricted to the corresponding layers as shown in the above figures. For example, excitatory neuron type 4 to 6 were localized in the layer 4, 5, and 6 in the above figure, and violin plots also shows the localized pattern of distribution in the layer 4, 5, and 6. Then the performance of spSeudoMap was compared with the two methods, cell dart and cell to location. The simulated subpopulation single-cell data was directly applied to both methods. The layer discriminative accuracy of each excitatory neuron fraction was measured by AUC, values of ROC analysis. spSeudoMap was superior to cell dart in 8 out of 10 cases and superior to cell to location in 7 out of 10 cases. Overall, spSeudoMap was successful in predicting layer-specific distribution patterns of 10 neuron types. Next, spSeudoMap was evaluated in mouse brain tissue. Among the cell types present in the single nucleus dataset, only the region-specific neuron types were selected, and they were utilized as a simulation dataset for the sorted single-cell data. When the main region-specific neuron types from the single-cell data were spatially mapped to the mouse brain using spSeudoMap, the spatial distribution of neuron types were highly localized to the corresponding anatomical locations. The anatomical regions were segmented by unsupervised spot clustering, and the clusters were named based on the Allen Reference Atlas. The outer, mid, and inner cortices, hippocampus, thalamus, hypothalamus, epiderma, amygdala, and amygdala piriform cortex are the main regions. Excitatory layer 2-5 were restricted to the mid to outer cortical layer, excitatory 2-3 to outer cortical layer, 5-6 to inner cortical layer, and the hippocampal neuron subtypes to the hippocampus and thalamus to thalamus, and piriform subtype to amygdala or piriform cortical area. This distribution patterns were highly similar between spSeudoMap and the reference results by cell dart. Then the spatial distribution of the rest of the region-specific neuron types with low cellular abundance were visually assessed. Many of other minor region-specific neuron types were localized to the corresponding anatomical locations or similar to the reference results by cell dart. Lastly, spSeudoMap was evaluated in breast cancer tissue. The sorted single-cell data from breast cancer patient was applied to spSeudoMap. The sorted single-cell dataset from the same patient was applied to cell dart, and the prediction result was considered as a reference for evaluation of performance. Of note, some immune cell types, such as effector CD8 T cells and basophils, showed mismatch between the two single-cell datasets. The spatial compositions of immune cell types were predicted by spSeudoMap using CD45 positive sorted single-cell data and visualized on the tissue. The results were compared with the cell type mapping results from CellDart using single-cell data covering all cell types in the tissue. Macrophage, CD4 T cells, and B cells showed similar spatial distribution between the two results, while CD8 NKT-like cells presented slightly different patterns between the two methods. Then, the correlation between the cell fraction predicted by spSeudoMap and that predicted by CellDart with whole cell types were calculated across immune cells. The cell fraction predicted by both methods showed positive correlation in major cell type macrophage. Memory T cell and B cell also showed positive correlation. However, CD8 positive NKT-like cells, which is minor cell population, showed negative correlation between the two. The predicted spatial patterns of minor cell population could be affected by immune cell type mismatch between the sorted single cell and spatial data. Finally, Spearman's correlation coefficients were calculated between the predicted cell fraction of representative immune types. The proximity of the spatial correlation patterns were examined and hierarchical clustering was performed. Macrophage and the T cell populations showed high similarities among the cell type pairs, which implicates the high interaction between macrophage and T cell populations in breast cancer tissue. To summarize, we developed spSeudoMap, which flexibly predicts spatial composition of a few cell types using unmatched single-cell datasets such as sorted single-cell data. It utilizes sorted single-cell data to generate the synthetic cell mixtures that closely resemble the spatial transcriptomic data and trains a model to predict spatial cell compositions using domain adaptation. The main idea is to assume the cell type that is exclusively present in the spatial data and assign its proportion and expression profiles based on pseudobulk transcriptome analysis. This approach, named spSeudoMap, was applied in brain tissues, and the simulation dataset containing region-specific neuron types could be accurately mapped to the corresponding locations. Moreover, cell types defined from sorted single-cell dataset of breast cancer were mapped to the tissue with spSeudoMap, and the complex immune cell landscape could be investigated. We expect spSeudoMap to enable the flexible application of publicly available cell sorting datasets and shed light on spatial dynamics of a few but crucial cell subtypes. I greatly appreciate Professor Hong-Yun Choi and Dong-Soo Lee for introducing me to the field of transcriptomic analysis and teaching me throughout the whole research project. This is the end of my presentation, and thank you for listening.